welcome you all to, this is kind of a special seminar series. This semester hasn't started yet, it will very soon. But we had a large meeting here in Miami for the American Statistical, well, the Joint Statistical Meetings. And we're fortunate that Joel Bader, who's our speaker today, also spoke at the conference and has extended his stay an extra day so he could come visit us and give us a talk. Really interesting background. It shows where sort of this, well, the statistical field in a sense is kind of broadening really nicely. So Joel's got a bachelor's degree in biochemistry from Lehigh University, and then he was at Berkeley where he got his PhD in theoretical chemistry, and then did a postdoc at Columbia also in theoretical chemistry. He's had some sort of a mix of positions in industry as well as academia. So from '95 to 2003 he was at a place called CureGen Corporation, where among other things he was director of bioinformatics for a good number of years there. And since '03 he's been an assistant professor in the Biomedical Engineering Institute at Hopkins. Really kind of broad range of interests. So I've grabbed this from his lab webpage, but sort of three broad areas that he describes in terms of his research interests. So he provides, or his lab provides, bioinformatics and computational expertise for a large-scale experimental effort where they were looking at protein wiring diagrams for Drosophila, and that paper actually appeared in Science in 2003. The lab now is working on a similar kind of map for human data as well. Another area that they're interested in is developing algorithms to infer topology of process-specific biological networks by merging information from full genome RNAi screens with data from large-scale screens for protein-protein interactions. And the third major area that they work in is understanding evolutionary motifs in genetic interaction networks, and they're looking for a redundancy in biological pathways by doing so. So it's a real privilege to have him here, and we're really excited to hear what he's got to say. Today, network diagrams in the development of disease. So welcome, Joel.
It's really great to be here. Thanks for the invitation. So I'm going to talk about two topics. So the first thing I'm going to talk about is our work trying to understand what's happening inside a cell as it undergoes some process. So instead of talking about this picture, I mean, it's a biological audience. So really, the idea is we want to know how protein complexes change as a cell differentiates into a new cell type. And that's an obvious problem to tackle, and for some reason, not so many theoretical groups, computational groups have been working on it. So I'll tell you about our efforts to understand this very basic thing of what changes happen to protein complexes during biological processes. And then after I talk about that, I'll talk a little bit about our work on genome-wide association studies. We have a new paper that just came out in PLOS Genetics on gene-based tests. So that was one of the talks that our group gave at JSM, and I'll tell you about our results there that I think are really exciting. So I'm really happy with that work too. So I should say, so a third new area that we've been working on is synthetic biology. We have work that's just been accepted by Nature to put a synthetic chromosome into yeast. So it's a collaboration with Jeff Buca's lab at Johns Hopkins. So if you want, you can ask me about that too at the end. All right. So here's what we're trying to do, is we're trying to get a moving picture of the cell where we want to see, really basically what are the protein complexes in a cell at any point in time, how do they differ with protein complexes at previous points, what's going on. And the challenge of developing a picture like this is that no one data set tells you everything. So typically we have static data sets of protein-protein interactions measured under in vitro conditions like yeast, essentially in vitro yeast-2 hybrid, that's just a biochemical interaction, maybe protein complex pull-downs. So a lot of static information that is maybe averaged over lots of different cellular states. And then we also have dynamic data primarily measured by mRNA abundance, either from RNA microarrays or these days from RNA-seq, looking at transcriptional state of a cell. And so what we want to do is merge together sort of these static databases of interactions with dynamic data about which proteins at any point in time are actually present in a cell, and put that together into sort of a moving picture of what's going on. All right, so here's, so my collaborators are Yongjin Park at Johns Hopkins, who's a graduate student, and then previous stages of the work, Chris Moore, who's at the Santa Fe Institute, Scott Patterson, who used to be in my group, but left academics to join Council, which is a personal genetics startup, although maybe these days it's too big to be called a startup. It's a new company. So Council's business model is that they are testing parents and fetuses for rare, very debilitating genetic disorders, and then doing reproductive counseling. So earlier results of this work have been published as part of a RECOM meeting in PLOS One, Asia Pacific Bioinformatics Conference last year. The stuff in dynamics hasn't come out yet. And so as sort of a funny joke slide, maybe not joke. So a good analogy of what we're doing is Plato's allegory of the cave, where we're sort of in a cave looking at a data set that is a projection of something real that's going on, and based on the projection, we want to reconstruct real life that's happening outside. So here's the data set that I'll be focusing on for this talk, yeast metabolic cycle. So I think many of us have heard about cell cycle and classic yeast cell cycle data sets that have been published on so much that I'm bored. They bore me at this point. So we thought we would try something different. So yeast metabolic cycle is this really interesting process that happens that's commercially important when you're growing yeast in a bioreactor to, make things or just to ferment. So if you grow yeast in a bioreactor, so this is work by Ben Tu and Steve McKnight, published in 2005. So you grow yeast in a bioreactor. This axis is time, and this is oxygen concentration. That if you take out glucose, the oxygen concentration goes up because cells aren't using oxygen anymore; they're not respiring. And then if you add some glucose, all of a sudden you get oscillations in oxygen consumption. And these are oscillations that are the population level, not the single cell level. So all of the cells are entrained on the same cycle. There's really not good knowledge yet of what actually the signal is that causes them to get all entrained. There's a suspicion that it might be acetyl-CoA that triggers things. But what's going on is that half of the genes in the yeast cell are oscillating roughly a 3-hour period. Cell division is entrained with a 6-hour period. And what's going on is that in the oxidative phase, so down here where oxygen is being used, cells are making ribosomes and synthesizing amino acids. And then in the reductive phase, DNA is being replicated, and then the cell is sort of being recharged. And so the theory that these people suggested, or the hypothesis they suggested, is that that you want to reproduce, you want to replicate DNA under reductive conditions so you avoid oxidative damage. So that's a working hypothesis. So these oscillations will go on essentially indefinitely. So unlike cell cycle experiments where you lock yeast cells, or at least in the yeast cell cycle experiments where you lock yeast cells and then have a trigger that lets them all start cell cycle together. So in those experiments, the cells dephase after 2 or 3 cycles. In these experiments, the cells are really all locked with each other, and there's no dephasing. It'll go on for days until either you stop it or I guess your culture gets infected. So previous work did a lot of gene expression clustering and found clusters of genes that cycle together. But really, there may be 4 or 5 different clusters because everything sort of that happens at one of these phases, everything happens together. So we want to understand what happens during a wave of gene expression. Do entire protein complexes appear and disappear? Do complexes gain and lose subunits? Are non-cycling transcripts involved? Something that we learned from cell cycle is that CDKs, cycle-independent kinases, don't cycle at all during cell cycle, and yet they're critical. So if you just focus attention onto what's changing, say during metabolic cycle, you might lose information about some of the key proteins because they're just not transcriptionally regulated. And our hypothesis is that if we put in information about protein interactions, we can sort of group together. We can look at a set of genes. Yes?
You used the phrase
Yeah.
Can you explain what that means?
All right. So here's a picture. So here's a picture that this axis is time, and these yellow lines are transcriptional profiles for different genes. And it's just saying that so here, there's sort of less going on, and then at this time point, there's sort of a big burst of stuff being turned on, and a big burst of stuff being turned off. So I'm just calling that a wave. So these things all go on in a wave. These all go off in a wave. But these that all come on together in sort of this narrow time point, gene expression clustering puts them in one cluster. Can we use protein interactions to say, all right, these all come on together, but these are really part of this complex. These are part of another complex. So can we break apart this big wave?
You want to cluster the part where it's really active.
Yeah, yeah. Maybe like adding protein interaction information.
Okay.
Do you see? Does protein expression also have that similar? Like, does the
So we wish we could measure protein expression. What we are doing now is we'll get to the end. I'll show you some of our specific predictions that now we're going to try to validate by actually measuring protein abundance with GFP tags or with pull downs or things like that. But there aren't. I mean, doing protein expression proteome-wide is not really feasible right now. All right. So that's what we're trying to do. So here's what we're going to do: is we have this static map of interactions with large data sets for yeast, for human. And then dynamic state, we have RNA profiles at different time points. And what we want to do is say at this time point, we'll sort of filter out the interactions that are present and then cluster those into protein complexes. And then we'll do that for the next time point and the next time point. And then we can sort of put it all together in a picture of what's happening to a specific protein complex that we've inferred, and then try to learn something. Oh, sounds like someone might have had a question.
How many protein complexes are there?
So we end up inferring about 300 protein complexes with a mean of about 10 proteins per complex. Looking at the data, I can tell by eye which ones really look right and which ones aren't really right. This is an algorithm. So we are not sort of building in a database of protein complexes. We're trying to learn the complexes from the data.
Right.
And we learn about 300, and we see that some sort of persist, some come and go.
And just to recap, at time one, what does the dotted arrow mean? You're finding which protein complexes are present.
So here's what this sort of means. So I'll get to how we generate these pictures, but each of these little circles is a different protein,
Okay.
and then the different colors tell us sort of abundance of transcript that we're using to infer presence or absence of protein.
It's a proxy.
Yeah, and so here's low expression, here's high expression. So red is sort of hot, so it's turned on. Blue is cold, and it's turned off. So this is, and then there are 36 gene expression snapshots that are roughly 12 snapshots for each 3-hour period. So here's sort of the first period, then the second period, and the third period. So you can also see how reproducible the gene expression data is, that the sort of the pictures are consistent from snapshot to snapshot.
So this is the same complex we're watching over time.
This is the same complex we're watching over time, although here, same.
Each circle is a complex.
No, each circle is a protein here. This is just one complex that we've pulled out. Yeah, and so here it's like actually most of the complex is gone. There are a couple proteins that are still being expressed, but most of it is absent. And then it gets turned back on during the oxidative phase, then it gets turned off again during the reductive phase, then it gets turned on and turned off and turned on and turned off. So here's the key mathematical idea. What is the probability of a protein complex? And so we're going to have a one-parameter model, and our parameter is going to be theta, which is a number between 0 and 1, that we say, given that these proteins are in a complex, what is the probability of a physical interaction between any two of the proteins? So it's a one-parameter model, and so the probability of the complex, given the edges, is the probability of the edges given the complex times the probability of the complex divided by the probability of the edges. The probability of edges are the observed data from the database, so we sort of just end up ignoring it because it factors out. And so what's the probability of the observed edges given the complex? which you could also call the likelihood of the complex. It's probability of edges given theta, and then being Bayesian, we're going to integrate out over this parameter. So theta is a nuisance parameter. So what's the probability of our observed edges given theta? So it's a really simple model. We'll say that for each edge, we have probability theta that the edge is there, and probably 1 minus theta that the edge isn't there. So in a complex with E edges, and then I'll use H for holes. So the absence of an edge we'll just call a hole. It's equal to the total number of possible edges minus the observed edges. So for this network, it would be theta to the 9, because we observe 9 edges, and then 1 minus theta to the 6, because there's 6 pairs that don't have edges. So that gives us a maximum likelihood estimator for the parameter theta, just the observed edges over the total. We can calculate the probability of the edges, maximum likelihood. But what we end up doing is we integrate out theta. So it's a parameterless model that gives us the probability of the edges given the complex, no adjustable parameters.
And all the edges are essentially independent.
Yeah.
In this model.
So that given 2 proteins are in this complex, the probability of an edge all the edges are independent, conditioned on they're in a complex and on the parameter theta that we integrate out.
And identical.
Yeah.
Yeah.
Yeah. That's right.
So any 2 proteins have the same propensity to
Interact.
Form an inter-
Yeah. So so there you go.
What is the 15 again?
Oh, so the 15 here is just that we have 1, 2, 3, 4, 5, 6 proteins in the complex. So the total number of edges that we can have is, 6 choose 2, or 15. So that's where the 15 comes from. If we had 7 proteins, we would have had 7 times 6 over 2, whatever. All right. So now we can calculate the probability that it's 2 complexes. So color some red, color some blue. Say we split it here, and it's exactly the same calculation, except now that we have 3 parameters that we're going to integrate out. We'll have a parameter for the probability of edges on the left, a parameter for the probability of edges on the right, and a parameter for the probability of edges that cross between the 2. It's exactly the same math, and now we get a product of 3 beta functions. So it's exactly the same. So this actually lets us choose when we look at a graph whether it's better describing it as 1 complex or 2 complexes. And so you can immediately sort of get the idea that we could do sort of recursive partitioning, choose the best first cut, break it into 2 parts, choose the next cut, break it in. There's one sort of last piece to think about, which is the probability of sorting proteins to left or the right. So we can handle that by introducing another parameter, pi here, that is the probability that that a protein sorts to left or to the right, and then we get the similar looking factor that we're going to sort n left proteins to the left and n right proteins to the right, and we can get a maximum likelihood estimate, or we can integrate it out. So we have another parameter-free model for the probability of any of the subsets of cutting a complex to left or the right. And so the probability, sort of our prior probability based on model space, where model space is sorting proteins to left or the right, says that putting everything in 1 complex is more likely than splitting things apart, because we want our model space to be normalized to 1, and we're just saying, we end up putting more weight on keeping things together than on splitting things apart. So that this piece is trying to keep everything together into 1 big complex, but the previous part of the edges is trying to push things apart, and together they fight each other until we reach equilibrium. Which, yeah.
So a little bit different from our part. Usually you have a variable. So you think about left and right in terms of the variable. What is left and right here in terms of the complex?
Oh, so we're just saying we have 2 complexes, and left is this complex and right is this complex.
With respect to what orientation?
Oh, we're not. Okay. So we're not talking about a physical 3-dimensional picture. Think more about graph partitioning. We're saying these proteins are in this complex, these proteins are in a different complex. So that's.
And you look over all potential partitions like that, or there's a particular one that you're zeroing in on?
So what we're trying to do is we are trying to find the optimal partition. there's the work that motivated us was a paper by Newman, Clauset, and Moore published in Nature in 2008, where they were doing Monte Carlo search using the maximum likelihood probabilities, not these fully Bayesian probabilities, to do Monte Carlo search. And we tried that, and you can't really go above graphs with 50 proteins in them.
Okay, let me ask my question again. So
Yeah.
the analogy here would be a categorical variable versus a continuous variable in the R-part paradigm, right? Is that the way to think? So you're thinking about cardinality.
A series of recursive binary cuts.
Yeah, in terms of the cardinality. So 5 pieces go over here, 1 piece goes over here.
Yeah. So now, as I said, you can do model selection, so we can compare 2 models and just calculate the probability of this model, the probability of that model, and you know, take the ratio and decide whether we should accept or not. So this would sort of be a Monte Carlo idea, accept or reject.
Is it kind of like a Bayes factor?
It's exactly a Bayes factor. Yeah. So you could also, instead of, you can also calculate the probability of even if you decide that the model says they're better separate, you can still calculate the probability that they really should go together. So you can calculate co-membership probabilities like that. Something else that's nice is it gives us an automated stopping criteria. So, so both at the bottom, so say that we're going to run this as recursive partitioning, and like any hierarchical clustering, typically you run exhaustively until at the very end everything is its own cluster. But here we can sort of say at the bottom we're really splitting on noise. Let's use these Bayes factors to decide below this point everything's noise, let's collect everything back up. But also top down, you can also find clusters that are really distinct from each other that really don't merge in together. So, so completely sort of disjoint pathways in a cell. It might not make sense to merge them together. You might want to say you have a bunch of top-level groups. So we also have criteria for stopping at the top just by saying it's better to keep these separate than to say they merge together into some sort of hierarchical pathway. So what we'll typically see when we run this is that if there are clear subunits, like proteasome large subunit, proteasome small subunit, or ribosome large particle, ribosome small particle, those things will sort of be our bottom level groups. And then above that, we'll go to merging the large and the small subunit, because there's still interactions between them. We might merge a couple levels more, and then we'll stop, and we'll say this is sort of a hierarchical decomposition that is interpretable and makes sense. But there are no adjustable parameters. It's all done using this Bayesian stuff. So here's a picture of what's going on when we apply this to yeast, where this is pulling out the entire sort of everything annotated as protein transport comes out as one of these top-level groups. And then in different colors are the bottom-level groups that our model detects. So this sort of group we say is one bottom-level group. And then the other slides, or the other panels, I'm highlighting what gets a Go category annotation. So Golgi to plasma membrane transport. So Go says this is sort of the bottom level category, but our method sort of pulls out this as a separate subgroup and these as a separate subgroup. These that are gray are probably part of the purple subgroup, but just not annotated yet. So we have a different level of resolution compared to Go. We think it's probably better. But it's also sort of cross-cutting in a little way that Go will group things together that so based on our method we would say really you can think about as separate. So now one of the tricks in evaluating methods like this is we want to compare how we do for classification versus the truth. But we don't really trust this truth. So the way that we think is really best to evaluate different method performance is that if you think back, we're using these parameters that describe the probability of a protein interaction. We integrate them out, but we could still get a posterior estimate. So you can actually use that for link prediction. So the idea is take out some of the edges from your real data set, then cluster the data set. The clustering sort of induces predictions for connections, and then see how you do on predicting the connections that you've left out. And our method works best. So we think our method works best. We compare it to a bunch of other methods, and and our method seems generally to work best. There are other clustering methods. This blue line is not a clustering method. The blue line is Google PageRank. So, okay, so what is each of these is a precision recall curve. So actually no. So these are so all right. So the top data set is for the top panels are for protein-protein interactions. The bottom panels are for genetic interactions, and then these are precision recall curves. These are F1 scores, and these are AUCs. And I'll just focus on these two because the precision recall curve really is, I think, the best way to think about it. So what this is this is precision and this is recall. So it shows how you start with your first predictions where you're making a really precise prediction for links, and then as you make more and more predictions, your quality goes down. And so here in black is our method, and then the next best method here is PageRank graph diffusion kernel that does well until about here, and then it starts to fall off.
Is that a clustering method?
No, it's not a clustering method.
What kind of method is it?
So graph diffusion, it's PageRank, which is essentially repeated matrix multiplication. So it's essentially counting up first neighbors and then second neighbors. So let's see, between each two proteins, you count how many length 1 paths there are, and then you add on how many length 2 paths there are, but with a decay factor. Then you add on the number of length 3 paths, but with a bigger decay factor.
So it's a weighted assessment of connectivity of the graph.
Yeah, that's right. Yep. Yeah.
So the idea so this is a cross-validation kind of thing that you're doing here, and your validation set are links that you've randomly taken out.
Deleted. Yep.
Okay.
And it shows how well we recall them.
And so you build the complex together on the 80% that left is in your training data.
That's right.
And from that, you're supposed to be able to predict
the ones that were left. Yeah. So basically, we rank them all, and then we rank together. Every pair that doesn't have an edge between them, we rank them in decreasing order,
right.
And then we look to see which of those were actually the real edges that were left out. So also something about this that's nice is it extends to multiple edge patterns. So say that you have a lot of crosstalk between like receptors and ligands. So you might want to group receptors together and ligands together, even though ligands don't interact with any other ligands and receptors don't interact with other receptors. And so that's sort of this picture where you might see some that based on interactions between receptors and ligands, you could group receptors together and ligands together. And that happens because these theta parameters that describe edge probabilities, they can describe a condition where inside a group you don't see edges. So it works well for other types of networks. Instead of describing those results, I'll describe a surprising conclusion is that right now there doesn't really seem to be so much synergy. So we thought this would be great for combining information from physical interactions and from epistatic genetic interactions, but unfortunately, we didn't really see such great synergy. So when we predict protein interaction edges based on protein interactions, that works better than predicting protein interactions based on protein interactions plus genetic interactions. And our suspicion is that the reason for this is that it's sort of somewhat disjoint sets of proteins and genes that have been studied by different methods, that the large-scale studies of genetic interactions have focused on a different subset of proteins than those studied for physical interactions. So it's a little disappointing. So I don't think it means that either the method is bad or that the data is bad. I just think it's just that there's no synergy yet. And
how is a gene-gene interaction defined?
So this is mostly synthetic lethal experiments looking at the phenotype of a double knockout compared to the phenotypes of the 2 single knockouts, primarily whether 2 single knockouts are viable and then the double knockout is lethal. So here's so intuition about some reasons why our method performs better. So something that's sort of interesting is that you can think about a flat. So often when you think about clustering, you think about a flat model where you're putting things into disjoint groups, but without any hierarchical relationship between the groups. And in a flat model, if you have k groups, each group has a pattern of interactions with all the other groups, and you have k-squared parameters, as opposed to hierarchical models where you have a branching tree, and to figure out the interaction probability between group 3 and group k, that's a parameter that sort of corresponds there. And that would end up being the same parameter for an interaction with this group in group k. So you have far fewer parameters. And so we think one reason why flat models don't perform as well is just they have too many parameters. And hierarchical models cut down on parameters and seem to perform better. We've had some so thoughts about different optimization methods. We've actually tried all of these. Monte Carlo, way too slow. We actually tried variational methods. The problem with variational methods is that we find that if you have a big network, you need a good starting point or you converge to a poor answer. And so we started with the work I'm describing by saying, all right, let's figure out how to get a good starting point for variational. And then we just didn't want to do variational. And then deterministic is sort of the type of algorithm that we're working with. And so one of our insights that I haven't seen other people talk about, but is actually the thing that saved us, is thinking about clustering as forward regression versus backward regression. So here's how people typically think about clustering: is you have a bunch of vertices for us for our work, proteins that you want to cluster, and you start them sort of all like a star graph. So basically, there's one parameter that tells you about the probability of interactions between any two pair you pick. They all share the same parameter. So it's a one parameter model. And then you're going to sort of break off two and say these have a special pattern, but then everything else just has the same single parameter. So every time you merge two groups, you're adding a parameter for merging two groups. So that's the forward regression picture. So here we've merged two groups, we've added a parameter, so sort of each internal node is a new parameter. So that's the standard way. The way that actually works much better is backward regression, where we start with, if we have V, capital V for vertices, genes, we have V times V plus 1 over 2 parameters, because every pair has a parameter. And then when we merge two groups, we're actually getting rid of a lot of parameters, because when we merge them, we say that instead of having this interact with everything else with its pattern and this interact with everything else, they're going to share a parameter. And then that knocks out the number of parameters that sort of scales with the number of groups. So we're losing a lot of parameters every time we merge, and it ends up working much better. And then there's actually, using all this Bayesian stuff, there's actually a lot of pressure to remove a parameter. That every time you remove parameters, you get a big benefit. So it works much better.
Does it work better in terms of like it gets you to a better answer?
Yes.
Oh, okay.
It gets you to a much better answer.
Computational issue is it?
Computationally, it's slower.
It's slower.
So I would say that's the drawback, is it scales one level worse with either edges or vertices. So this method that, as far as I know, I haven't seen anybody else use, it scales to about networks with, I would say, 100,000 vertices, maybe 1 million vertices if you have a really good computer. For genome scale, it's completely fine. So it's the sort of thing that you can do for genome scale, but that you might not do for large, like for Facebook or phone call records with 1 billion vertices. I think you'd have trouble, but I mean, maybe there's some heuristics you could do first. But it gives you just such a much better answer. And so I'll tell you the insight about why it gives a better answer. So it turns out, like with this forward approach, if these two share an edge, basically, or interact with each other, basically the forward approach, you get pretty much the same benefit by merging together any two proteins that interact. What this approach does is, in addition to looking at whether they interact, you also look to see whether they have interaction partners in common. So this way of doing it, instead of just looking at edge sharing between these two groups, you also see whether they have the same pattern with everything else. And that draws a lot more information. The drawback is that you have to look over all of these groups. So it gives you much better performance at the cost of sort of an order of algorithm of an extra order of the number of groups or the number of vertices or the number of edges, but it works so much better. All right, so that's for dynamic data. And so all of that has been published. And now, so this is the unpublished stuff that we're currently working on, is that now we want to look at a network that's evolving over time. And we do it with a path integral or hidden Markov model formulation. And so maybe you're familiar with hidden Markov models from protein sequence, where you have some hidden state, and then at each state in the Markov model you emit an amino acid. There's several talks about doing this for copy number variation, where your hidden state is the number of copies. So here, our hidden state is this sort of definition of the group structure of a network. So our hidden state is our assignment of proteins to complexes, and then the hierarchical organization of complexes. And our emitted state is a G for a graph. So we observe graphs that are emitted at subsequent times. And basically, the way that we're getting those is we are taking our full database of protein interactions, and then we are just projecting out the subset corresponding to proteins whose transcripts are present. So we have a graph that changes with time. And so our joint probability over this whole trajectory is the probability of our initial state, and then the probability of the first emitted graph given the initial state, then times the probability of the first of the model at time 1 given model at time 0, and then the probability of model at time 2 conditioned on model at time 1, the probability of graph at time 2 given model at time 2, and then sort of the next transition, and the next admission, and the next transition, and the next emission. And
so G on the left is
the full trajectory.
Oh, it's a full trajectory. So
All the
it's
G's.
Yeah.
a vector of G1
Yeah.
and GK or something.
That's right. So this is the joint probability of all of the emissions, so all of the observed networks, and all of the clusterings. Yeah. So it would be like, the joint, yeah. So it's just the joint probability of everything, of the observed data and the hidden model. And then we're just using Markov properties that conditioned on, so the observed networks are independent conditioned on the model at that time. And the model at one time is conditionally independent of previous stuff, except for the
M is the corresponding vector for
Yeah.
M naught, F1,
Yes.
M2,
Yeah.
and so on.
So to account for dynamics, so these parts, this is what we just, the previous part of the talk is clustering a single snapshot. And then these parts, so you can think about as this is the joint probability of clustering at time 1 and clustering at time 2, divided for normalization by the marginal probabilities for the clustering at time 1 times the probability of the clustering at time 2. And so again, we do this all Bayesian. What we keep track of are proteins that are in complex K at time 1 and complex K prime at time 2. And then we have a nuisance parameter for the probability of a transition like that. And so then we have the joint probability conditioned on theta, and then we can integrate it all out, and we get sort of ratios of multinomials. So it's a parameter-free model. Yes?
HMM, what is there?
You
HMM?
Excuse me? Oh,
Okay.
oh, I'm sorry, hidden Markov model.
Oh,
Sorry.
okay, you said too fast.
Sorry. I'm sorry. But so also this sort of gets away from having to identify what complex at time 1 is equivalent to what complex at time 2, because we end up summing over everything the way that a good Bayesian would. Although that
But
sort
you're
of
really interested not in individual sequences G or M. You're interested really, aren't you, in sets of Gs? Because you're not going to have a unique trajectory, are you?
no, you're not going
So
to
you
have a
wanna
unique.
sum
We're summing over all of them. Yeah.
PGM over G and M in some set that represents a pattern of interest. Yeah?
So I'm.
You could think
It-
of the trajectory as a pattern.
An individual trajectory is a
Oh,
pattern,
okay.
and you want to take a sum over those patterns.
Yeah. So it's yeah. Although at each.
So this is for an individual sequence, but you're interested in sets of sequences, aren't you?
No, this is
for one sequence.
This is for, yeah, so this is,
Oh, no, I understand this is for one sequence, but I mean the physical problem.
so the physical problem is that we want to see how a protein complex evolves over time.
Yeah.
And at this time, it'll have some proteins assigned to it. And at this time, it'll have some proteins assigned to it. And, but in this math, we're not making a direct association of saying this complex at this time is identical to this complex at this time. Instead, we're sort of summing over all possibilities, all possible connections. And in the end, we could calculate posteriors and calculate like for, what's something's best match. So we could calculate, and I'll actually get to that a little bit. So essentially what we do is first we cluster each snapshot, and then we sort of recluster based on the model to the left and the model to the right. We can use those as like influence functionals to tell us how to recluster the one in the middle. And then that makes the clustering sort of consistent over time points, and we iterate to convergence.
How dependent is this on M0? Where you start?
We take an uninformative M0 so it ends up not really depending on M0.
Okay. I'm just saying, like, if you pick different M0s, do you get like vastly different trajectories, or is it?
So we haven't. I think our M0. I forget whether it starts with everything in one complex or everything together.
So you're trying to pick
But-
something sort of non-informative.
Yeah, actually, no. I should essentially this doesn't. If we start here, if this is our first data point, essentially this sort of goes away,
Okay.
because we're going to marginalize over M0, and it turns out that it just sort of goes away. So we would start with the probability of M1 times the probability of G given M1, and then the probability of M2 given M1, and then that probability of M1 is just that multinomial that we have with a single snapshot. So essentially, that's what happens. So then we have the matching problem of tracing a protein complex over. And so the way that works is, so we can give, so like we want to say, all right, this complex at this time point is equal to this complex at this time point, and that's what happens with it. And we can weight each of these edges by the probability of, the probability of transition. So this is essentially this essentially ends up being the number of proteins that are shared by the two complexes times the log of the number shared divided by the number expected to be shared. So it's that sort of thing. So we get a weighted network, and then we can do it with a minimal spanning tree, with Viterbi to choose the maximum weight path, then subtract it, do the next path, maximum weighted matching, that also works. All the methods work pretty much. They all work well. The real problem that we have is sometimes a protein complex goes away entirely. So in these data sets, if a protein complex goes away entirely and then it comes back, we sort of have these threads that are broken and we have to match across. So we have a bunch of hacky ways to take care of that. We don't have a fundamentally good way. And the reason is the Markov property, because this is all a Markov model based on the preceding time, and if something goes away, then you have to look back previous time steps. And so that's really what we have to bring back into it to get that to work. But still, so here is pictures of what's going on. So back to the real data. So for sort of a best matching, this graph shows the consistency of complexes based on Jacquard coefficient between pairs of snapshots. And so usually it's up about 95%. So when we say two complexes are the same, over two time points, they'll share about 95% of their complex members. But then when there's one of these waves of gene expression, the similarity drops down a little bit and then sort of comes back up. Then it drops down sort of at the next big wave of gene expression. So that makes sense. Here's a global view of these complexes that we get, where here this axis is time from 1 to 36. And then each of these rows is a different complex that we're sort of tracing from one time point to the next. And then I'm zooming in here. So the green color is a complex where protein interactions are enriched. The blue color is complexes where interactions are depleted. And I should call these groups of proteins instead of complexes, because really, looking at this is really just noise, I would say. It's a group of proteins that we put together with sort of weak information when you look at it. I don't know if it's really credible, but these green ones are highly credible. So I think one thing that we need to do is have some sort of quality that lets us know, but I can tell you it's very easy looking by eye at the results, which ones to believe and which ones not to believe.
So
And
what's the size?
the size is the number of proteins in the complex.
Okay.
Yeah. All right. So now here are some examples. And so now what I'm going to do is zoom in on one of these threads and then sort of blow up what happens at each of the snapshots. So here's the one from the beginning, boring regulation in the ribosome, that it's expressed during oxidative phase and downregulated during reductive phase. So here it's sort of medium on, medium off, and now it's sort of like all the way off, and then it's sort of at a low level, and then it comes back on really highly, and then it goes back off again. And it's really, I think it's sort of really beautiful data. And also, so something that I think is interesting here is that to do an analysis like this, you probably don't need to worry about replicates of RNA so much, because you sort of, if you are sampling expression dense enough over time, just your replicates essentially are the previous time point and the next time point that that sort of smooths things. And also, we're looking at the level of complexes, not at individual genes. So that sort of, I think, does some noise suppression. All right, so here's exciting regulation, the ribosome. So it's the same picture exactly, just a different title. And what's exciting, or at least intriguing, is that if you look, there's this protein colored in red that comes on when everything else is blue. And then when everything else is red, this one is blue. So so that's our hypothesis, is that it's a regulatory protein, and so it's RPS6A. It's a regulatory protein in the 40S subunit. It's part of the TOR signaling cascade, and actually dephosphorylation leads to growth arrest. So you can imagine that when it's newly synthesized, it hasn't been phosphorylated yet, and so it's sort of turning off activity of the ribosome, and then it sort of, then you turn off its expression and it gets phosphorylated and things turn back on. So we're trying to validate this in two ways. One is doing protein complex pull downs to see that the stoichiometry changes or is time dependent. And then also there's work going on in Ben Tu's lab looking at acetyl-CoA as a sensor that regulates activity of histone modifying enzymes. So then there's some other examples like this. So here's something called the GID complex, and there's a protein that's sort of at low levels, or transcript that's at low levels when everything else is high, and then when everything else is low, this is high. And then sort of another antiphase example, RNA maturation. So what we found with this method that seems to be sort of a new story is antiphase expression of proteins that end up downregulating the expression of everything else in the protein complex. So that's what we've learned so far. So we have a couple other studies in process using this method. Differentiation of dopaminergic neurons, which is actually the problem that motivated our work. There's a gene called HPRT whose mutations cause Lesch-Nyhan syndrome. It seems to involve a failure of neural development, and we're collecting, well, our collaborators in Ted Friedman's lab are collecting RNA-seq data for a 2-week differentiation of induced pluripotent stem cells, and then we want to see what sort of is going on, and then we'll repeat the experiment for cell lines that are defective for HPRT. And then we're in a collaboration with Sasha Popel at Johns Hopkins looking at angiogenesis. So looking at gene expression changes as cells start going through angiogenesis. And we're going to release our software under GPL. So your study too. Or if you want to ask Youngjin Park over there, who is here also, maybe he'll run your data set. But we're really interested in trying this out on other data sets. So Youngjin Park, and then funding for this is primarily through NSF and an NIH Roadmap Center. And so now I'm going to give, so I heard that, so now a special quick bonus talk from JSM. All right, so I won't go past 11:30. So just in case you don't care about systems biology, but you care about GWAS, I'm really excited to tell you about a new GWAS method that we've developed with Don Arking, also at Johns Hopkins, primarily students Hailong Wang and postdoc Pranam Chanda for this problem, where you've done GWAS, you've done SNP-by-SNP analysis, and red and blue here are two different LD blocks. And both LD blocks have sort of reasonable signal, but not genome-wide significant. But your idea looking at this is, oh, these are two signals in the same gene. If I combine them, that should be significant. So I want to do a gene-based test. But the problem is that, to our knowledge, there aren't really any good gene-based tests that overall have better power than SNP-by-SNP analysis. So the related problems say that you have one hit in a gene that's genome-wide significant, but you have other hits that aren't quite. Do you believe the other hits conditioned on seeing the first hit? So that's a similar problem. Then ideas about meta-analysis. What if different genes in different populations, and what if the same gene in different populations, the best hits are a little different? If you're doing SNP-by-SNP analysis, you might miss that. If you did meta-analysis at the gene level, you might do better. Also, gene-based p-values are better for integrative analysis where things are really based on genes. So here's the spectrum of what people are doing. So here are the number, like you look at a gene, how many SNPs do you put in your model for association? On one extreme is just taking the best SNP in a gene, which if you're not going to use our method, our recommendation is to use just the best SNP in a gene, possibly corrected for the number of SNPs in the gene overall. There are methods that say every SNP in a gene has an effect and sort of sum up the effects over the gene, either summing chi-squared scores for every SNP or summing up Bayes factors. These methods aren't quite as good as this method. Bayesian Markov chain Monte Carlo, that's just too slow. No one ever would do that. Despite, I'm sorry, I hope I'm not insulting anybody. But, all right, despite talks at JSM to the contrary, no one ever does Monte Carlo genome-wide. And then Lasso, which is really fast and really popular, but I'll show you, in our hands, was really one of the worst performing methods.
So why does the best SNP method work better than the sum?
Oh, so the best SNP in a gene says essentially your hypothesis is that there's only one effect in this gene. All right? The sum method says essentially I'm summing up chi-squared. That means every SNP in the gene is really functional. And if the truth is closer to there's only one effect in a gene as opposed to everything, every variant in the gene has an effect, then you're sort of close, even though this isn't Bayesian, you're sort of close to essentially the places where this hypothesis is, the null. Yeah. So that's entirely why. And actually, there's another reason why these. I shouldn't say there's. That's really the main story. Is that if the truth is there are a small number of effects, then a method that's really testing against hypotheses that there are a small number of effects will do better. So our motivation is really Bayesian, but here's sort of most of the story is you can think about, Bayesian you're usually sampling. Say instead you start from a null model and then just do greedy forward selection based on the Bayesian, the Bayes factor. Put the SNP into the model that best improves the Bayes factor and keep doing that until the Bayes factor says stop. That's all we're doing. No one's done that before to our knowledge, and it works great. So this is just the standard Bayesian stuff for calculating the probability of association given no association. You're really supposed to sum over all models other than the null and then normalize by the null. Instead of
some. I think the forward searching, because the paradigm in regression is that doing just forward searching kind of sucks.
That's because, all right, so here's why people say it sucks, and they're right. If you do that, what's your criteria for what model to stop at? And typically people say, oh, I'll use BIC. And that penalizes for the number of features you've put into the model, but it completely ignores the number of features that you chose from. So, and also, it's not just the number of features you chose from, it's also subset selection. Because really, it's not, you're choosing one particular subset of features to put in. So what you need is a prior on feature space. And so that's this thing. What's your prior on feature space that's normalized? And there are standard priors on feature space to use. And I'll show you the prior on feature space that we use. So one thing is that you can't do the full sum. So instead, we're just going to try to approximate the full sum by just the best model. And then we can't even get the best model because that's an NP-hard problem, so we'll do this greedy thing to try to find what the best model is. So here's the prior unfeatured space. So this is really the same math as in the first talk, that we have k SNPs in a gene that we're going to put into our model, and then t minus k that we're not going to put into the model. And let's just say there's a parameter theta that's our prior probability that a SNP in a particular gene is functional. So theta will vary from gene to gene. So, but we don't know what theta is, so we have to integrate it out. So it's a parameter-free prior. We just take uniform. We get this actually should be k plus 1, t minus k plus 1 instead of t plus 1. But anyway, that's our prior on model space, which is really the standard Bayesian prior. There's only one trick with this, which is when you think about SNPs in a gene, often things come in linkage blocks. So you have a block where lots of SNPs are correlated. And we want our method to sort of be independent of linkage block size, or like how many SNPs are in a linkage block. So instead of thinking about number of SNPs, we'll think about number of effective tests. And there are lots of methods that people have proposed for calculating effective tests. Like you would imagine you could do PCA and count up, number of eigenvalues. That works somewhat. We actually do what's like non-negative regression to figure out how many tests there are. Doesn't really matter so much as long as you realize you should be looking at effective tests as opposed to number of SNPs. And then this is just, even though it looks sort of complicated, maybe not, it just, the rest is just standard Bayesian stuff. And then, so here's what we're doing is we're selecting models based on the log likelihood from the maximum likelihood estimator, plus the BIC penalty, plus this log beta multiple testing correction, essentially. So we assign SNPs to genes. We have 20 KB flanking regions based on results from EQTL studies. Our results are not, so this 20 KB is actually the only adjustable parameter in our method. And when we choose different sizes, it doesn't really matter. And then we compare to other methods. And then we, so then the one thing that the computational cost of our method is that inside each gene, we calculate this test statistic. And then we have to do gene by gene permutation studies to convert it to a p-value that's uniform under the null. So our p-values are uniform under the null. It turns out like the people we work with, they've spent maybe $1 million collecting the sample and genotyping. So an extra $1,000 for doing the permutation test, it's like, even for the univariate SNP-based tests, they always do permutations. And our permutations don't take any longer than the SNP-based permutations. So really, and the time for this test is indistinguishable from the time for a standard test. So so one big difference between Lasso is that we have this L0 penalty motivated by Bayes versus Lasso's L1 penalty that's motivated by you can do it fast. How does it? So, and then instead of doing simulation studies, we care about performance in real data. So we're working with data from the CHARGE consortium, which is a huge cardiovascular health data set. So this is the data set that has led to most of the publications about ECG parameters like QT, QRS, PR interval. Right now, what we analyzed was, what's been analyzed in meta-analysis, 50,000 individuals, 2.5 million SNPs, about 1.5 million of them mapped to genes, 38 true associations. And our test is going to be looking at a smaller data set with only 8,000 individuals, how many of the true associations can we recover at genome-wide significance? So this is in practice what someone would want to know.
So you're kind of assuming that the Nature Genetics paper, the ones that they found are actually true.
They are, yeah, but they are. I've been brainwashed. They are true. They've replicated. Most of them have replicated. Using these 5 times 10 to the minus 8th p-values, almost nothing has been a false positive. So these are the results from the last year or two. So this stuff, I really believe, maybe 5% of it is wrong. But
at these meetings, you heard a lot about SNPs with sort of weaker association, lots of that, so that wouldn't meet these.
No, these have weak associations, but they're genome-wide significant. So it's a variant that has a small effect on the trait that, if it were a relative risk study, it would be like 1.05, 1.1. So it would be a small effect, but genome-wide significant. And then people can argue about whether you care about that. In fact, I think we don't know yet, but it it gets you to a gene that's in a pathway, and these are all real findings. So so it's it's real. It's real. So this is just in table form, and then these are the results of finding genome-wide significant hits. So our method does best, and then the other Bayesian method does next best. So BIM-BAM is one of these more expensive Bayesian methods, but it misses stuff. The best SNP in the gene does sort of okay. Vegas adds everything up, does sort of okay. Lasso really does poorly. Here's an example of a gene where we do better that no other method finds. So this is actually a region where there are two genes that look like they each have effects, but because they're so close together, we can't distinguish whether it's in which genes the effects are really in. But so there are 4 effects in this region. None of them is genome-wide significant, but putting them all together, we get a p-value that's like 10 to the 6 smaller than genome-wide significant. So we really pull it out.
Wait, can you go back for a second? On that plot, you have on one axis the minus log 10 to the p, and
Yeah.
on the other axis you have recombination rate.
Oh yeah.
Kind of describe, like, is there a relationship between those?
Oh, this was just plotting so that people can sort of see where LD blocks are.
Oh.
So it's like, so these are recombination hotspots. So sort of in here is what you would think of as maybe an LD block. And then this sort of shows that there are recombination hotspots here. So this is sort of an LD block.
Oh, I get it. Okay.
So it's a couple of questions. So you've got this blocking structure. So why would people not go to variants of the lasso that allow blocking structure like the group lasso or something? You to zero out things within the group as well as across groups.
What we've seen is that lasso does particularly badly on low frequency alleles. So I'll show you, and I think that no matter what you do with lasso, with the all right. So lasso, the way lasso works is you're penalizing proportional to the absolute value of the regression coefficient. Now say that you have 2 variants that have identical variants explained. Under a standard unpenalized regression, you'd get exactly the same p-value because they'll have exactly the same variants explained, and that's what's important for like the chi-squared. Under lasso, if it has lower frequency and same variance explained, it'll have a larger regression coefficient, and then lasso will hit on that regression coefficient proportionally more than on a common variant. So what lasso ends up doing is it ends up weakening the signal that you get from low frequency alleles. And so in this plot, I show power as a function of minor allele frequency for simulated data. And best SNP in a gene in our method are up here. So power is pretty much independent of minor allele frequency, which is what you expect based on knowledge of how linear regression works. Whereas lasso really does poorly. And Vegas, the sum statistic methods, also all do poorly. Principal component regression does poorly, and they do poorly because the higher frequency stuff ends up swamping the lower frequency stuff. And you could possibly think about fixing lasso by renormalizing allele frequencies, but then that causes other problems. So just in general, it seems like lasso, our perception is that lasso will typically have problems with low frequency. And also, lasso is more sensitive than L0 to having like a block where you have lots of SNPs.
So now
I would just suggest to add in the elastic net as a comparison.
that's basically the group lasso.
Yeah, because I mean,
We've done that, and it doesn't really.
Did it improve matters at all?
Not really. No.
So then the next thing is when you do these prediction games that people play with these things, right, where they leave out a test set,
Yeah.
and then they want to see when they plot y versus y hat for
Yeah.
the quantitative trait. What's the R squared of that?
Oh, so we don't look at that at all because the prediction quality, so predicting a phenotype is a completely different problem.
Right, exactly.
And we haven't worked on that.
Right. Because I've seen in like the animal breeding literature, some of these guys throw in all the SNPs.
Oh yeah, so that is actually pretty, so Peter Vischer has some papers where it's essentially a kernel where you're predicting a phenotype for somebody based on phenotype sharing, based on genotype sharing with other individuals. And that works pretty well. And it actually, I mean, it sort of tells you that there's really heritability there, even if you haven't found what's causing it. But that's a completely different problem that we haven't really tried to work on yet. We're interested in it, but we just haven't worked on it. But I would say, like for that level thing, probably lots of methods will perform well if you want to pick out the top set of features. So if you look at precision recall as your metric, then the methods all are roughly equivalent. So, but that's not, so the people we work with, that's not their assessment. Their assessment is, are you able to find stuff at genome-wide significance so we know where actually, we know which we should try to validate and which not.
Oh, I was just going to comment that it's roughly speaking, since the work of ZOU, that's Z-O-U, people haven't really, have seen that lasso is really only good in very, very narrow settings. And usually adaptive bird shrinkage methods and group shrinkage methods perform better. And what I was mentioning before about SCAD seems to be, from a predictive standpoint, as good as you're going to get, and it routinely beats, say, elastic net in a predictive sense.
In a predictive sense, or in identifying genome-wide significant features?
Well, I did simulations in a predictive context, so.
I don't know how it would do in like a GWAS.
In a model identification setting, I couldn't tell you.
So the next time we publish on this, we'll try to.
What I was gonna
See, it's like, but we're also somewhat limited by implementations that were available at the time, and there is a, there is a paper from in bioinformatics by Hasty in like 2009 that everybody was pointing to, and it's also, I mean, there are a lot of knobs to twiddle, and
That's correct.
Right.
That's correct.
Right.
And that's the knock against adaptive methods, because being adaptive, you have to estimate.
Yeah, and someone will always complain that you didn't twiddle the knob properly.
Yeah, and they could be very sensitive to the knob twiddling.
Yeah, and then if you really want a method that's going to be very sensitive to how you twiddle the knobs. So in practice,
But you see, that's what I was suggesting. SCAD has only one
knob to twiddle.
One knob to twiddle, but I think that for what you want, you probably want grouping, and so you want like an adaptive grouped SCAD, or my conjecture would be that an adaptive grouped SCAD.
Well, if someone publishes software like that we can just run.
SCAD is out there. The software for SCAD is.
All right, so but adaptive grouped? So I'm not familiar, so I'll have to take a look.
No, not I just made up adaptive grouped SCAD.
You see? There you go.
But you're right. But
No, so maybe it's a good idea. Maybe it's a good idea, but I mean, we're not. We're not going to try to implement something new to compare with, just to compare with a lasso-based method. But if there's a method that everybody is using and saying, it gives good results, then we're definitely going to compare with it.
Oh, no, I'm just arguing that it's not a surprise that lasso.
Lasso does poorly. Yeah, it was to the reviewer.
Oh.
Not if you get weak signal. I mean, lasso is geared for situations where there's sparsity and high signal.
No, yeah, definitely. But I think that hasn't filtered through to the GWAS reviewers.
Right.
Yeah, that's probably true.
So the question is that in these plots, not only for common alleles, for high frequency alleles, because of 30%, 40% is very high.
Oh yeah, so it does worse overall, but it does much worse. In the lower limit. Yeah. Yeah.
So how computationally expensive are some of the things that you're talking about? Like your networking, modeling, and your stuff. I mean, are we talking like cluster implementation?
So did I have a slide on CPU time? All right, so here's this stuff. So here are CPU time in hours for doing a genome-wide study on one CPU. So for null data, it's mostly between 10 and 20 hours on one CPU. For real data, it goes up to like 50. Oh, here's 100 hours for BIM/BAM, but we don't really recommend that. But most of the others, it's like 1 to 2 days on one CPU. And the reason why it takes longer for real data than for null data is because when we're doing permutations, and you have to do more permutations when p-values are better. So you have good p-values, it takes maybe twice as long. So this is very reasonable. And on the previous talk, that's like 1 or 2 hours on one CPU. So it's very feasible.
I find it kind of p-values and Bayes. Is there a logic for when you use one over the other?
We're using Bayes to get us a test statistic. So think like we're using Bayes to get us a test statistic, and then we want to convert the test statistic to a p-value so that we know whether, so that we can sort of have all genes on the same footing.
Bayesian multiple comparisons would be.
What do you mean by Bayesian multiple comparisons?
Well, when you have all genes on the same footing, Bayes testing doesn't do that. I'm surprised.
No, because it ends up being sensitive to gene size.
Sorry?
It ends up being a little sensitive to gene size. So we're not. So that's actually something that we haven't really. It would be very convenient for us if we had an easy calibration between this, Bayesian motivated test statistic and a p-value, but we're not convinced we have a good one that's uniform under the null, and that's why we're just doing permutations. But I think it's a good suggestion to really see whether it is, and our experience is just that we haven't, we've done some just plots looking at different genes, how we get a calibration with p-values, and the extreme value distributions seem to be a little different for different genes.
New question, if I may. In one of your slides, it was around 10 or 11, you were comparing your method to sort of graph theoretic type clustering.
So this is the previous talk or this talk?
Previous talk.
Previous talk.
Okay.
And I asked you a question. You said that you were actually looking at this weighted sum of how many steps you were away from a given.
Oh, okay, yeah, graph diffusion. Yeah, looking at graph diffusion.
Yeah,
yes.
yeah. And I wanted to comment that there's a well-established literature in graph theoretic clustering stemming from work in the 80s by Shi and Malik.
Mm-hmm.
And there's these.
Oh yeah, that's Metis. Yeah, we compare to that. It doesn't perform so well. And the reason, yes, and the reason why it doesn't perform well is that does really well for graphs with a low dimensional embedding. So those were methods that were actually motivated a lot by figuring out how to put transistors on chips, minimizing the number of cuts.
Yeah, it's a min-cut map.
And so like a lot of the spectral clustering stuff, and it does really well for graphs that have a low dimensional embedding, but it doesn't do well for graphs that look more like random graphs. So yeah, so those methods do not really perform so well for complex graphs. Was there anything else I still wanted to say? Oh, so one thing that's interesting is that we can go back and we can say for these genes that really, through meta-analysis, look like they're associated with an EKG parameter, how many do we think have multiple functional variants in them? And so maybe half to 2/3, we say there's really only one effect, but for another half or a third, there are multiple effects. So this we feel is why our method performs better, because it seems like the truth is that if a gene has one causal variant in it, it has a higher prior probability of having additional, which makes complete sense.
There was a talk, actually, the guy didn't show up, a guy named Daniel Shane.
Oh yeah.
He was talking about gene set analysis
Uh-huh.
of G1s. Yeah. So I went to it, but
So that's exactly the same idea at the pathway level,
yeah.
that if you have a pathway that has one gene in it with a real effect, there'll probably be other genes in the pathway. So I guess I'll just get to, through simulation, we tested that we're actually estimating the right model size. And we're a little conservative, actually. So our models are usually smaller than the true model, at least for simulated data. And that's even the case if we throw out the causal allele and we only have markers that are an LD with a causal allele, we're still conservative. So our model sizes we think are believable. And then lasso model sizes are just all over the map. So here's the real model size. So this is the true model. This is our estimated model. And then so this is where we are. So we're typically under predicting the real model. And then lasso just has a huge variance.
Yeah, it's probably on average a little more accurate, but the
Yeah, on average, the variances. So here, it's just not usable really. So what we're doing now is we're extending it to meta-analysis. And the trick here is that if you want to do, so our method really depends, it's multiple regression, it depends on LD between SNPs. In meta-analysis, we don't have LD between SNPs. So what we're working with is methods where we use one population as a proxy for LD in a different population. So we're working on methods like that. Thinking about extensions to things like gene set enrichment analysis or interactions or pleiotropy.
So go back a second. You put studies do not share underlying genotype phenotype data. Is it just because people are like, it's mine?
So there are a couple of reasons. So one reason is that that often, somebody's really cleaned the phenotype, and there are different definitions of phenotype between study. So it could be that the phenotype is not even exactly the same phenotype or cleaned up the same way between studies. Also, there are concerns about patient privacy. And I think there's a lot of, there's not clarity about what it means to adequately anonymize a data set. So even if you wanted to share it, you might feel like, I'm just not sure. I think I'll get in trouble for sharing it. And so part of it's too valuable, and part of it is to protect people's information. So so a sufficient statistic for our method would be SNP-SNP covariance matrix, but that's a big thing. So that's also something that people just don't share just because it's so big. But it would be easy to share a subset. So that's, you could just say, all right, for variants that have a reasonable regression coefficient, why don't you give us the covariance matrix for those for everything with R squared greater than 0.1? So something like that.
I think that the SNP covariance matrix would kind of be block sparse. Like, I mean,
yeah, yeah. But you don't even need all of it. You would just need it for the SNPs with a reasonable regression coefficient, because those are the ones that are, those are the only ones that go into the model anyway. But there's not software to do that automatically. So we're thinking, so we're applying this to charge meta-analysis, and right now we're using one Caucasian population as an LD proxy for all the others. We've developed a method to estimate LD from one population, which actually improves things. But what we could just do is write a little script so that they could, in addition to sending the summary statistics, also send an LD summary that then would plug into this sort of meta-analysis. But then there are interesting questions about meta-analysis. So like, as you're comparing genes, typically you do meta-analysis at the SNP level. Here, do you have a model where it's the same SNPs in different cohorts with the same regression coefficient or with different regression coefficients or with different SNPs? And it's not really clear what will work best because this is a new area.
So when you look at something like the Sardinia study, so those are those founder population kind of things, right? So is that what you're talking about from the island of Sardinia where they,
yeah.
The idea is there's not a lot of intermixing.
So that would probably, we wouldn't want to be using European LD for that.
Right.
But so here's where we're going with that. We have a lot of HapMap we have HapMap and we have 1,000 genomes. And the same way that people use those to impute genotypes based on genotype markers, you could say, I could use those maybe to impute LD. And it hasn't been something that people have worked on because they're, imputing LD hasn't really been done. But this problem is going to be common for any gene-based meta-analysis so like say that, you really think, whatever it is, SCAT works great, and it's not SCAT? No. But so here's the thing, it just shows that these methods have not really been used in practice. Because if someone really was using this in practice, they want to be using it for meta-analysis. And if they wanted to use it for meta-analysis, they would have exactly this problem, that you need to have LD for each of the subpopulations. So this is, so there's been a lot of methods development, but in reality, there aren't many methods that are used in practice, and by far, univariate regression is the method, and it's the method because applied to real data sets as opposed to simulated data, it's had the highest power. So so I'll still compare with it, but this is it's showing that this method actually, lasso type methods also just have never been used for meta-analysis, because if they had, they would have exactly this problem. And we haven't seen any papers or any work on estimating LD, which is the crucial part of the problem for applying fancy methods to meta-analysis. So all right, so using our method, we found a new locus for QT from meta-analysis. So we went back to, so these are the published meta-analyses for EKG from charge, and then we went back using our, our somewhat sloppy estimate for LD, we're able to find a new locus. It's in a potassium channel, the same family as other QT loci. So 2 hits, neither genome-wide significant, but and we're validating this now in the MESA population. So so here's Hailang Wang, the person who's done most of the work along with Pritam Chanda postdoc. And funding for this work has come from NIH, the Kleberg Foundation for Medical Research, and the Simons Foundation for Autism Research. Thank you.